The point of this video is to examine some of the techniques that can be used to analyze data when the data may be low signal to noise. And the low signal to noise is related to low count rates, which may be caused by analyzing a small area of a thin film that necessarily has low signal to noise. And this is an example that we see here from a sample where a molybdenum disulfide flake is measured using a stack of images, and the images themselves will take a considerable length of time to acquire, but nevertheless, because we're analysing a small area and a thin film, that the signal itself is relatively poor compared to the noise. If we'd used a bulk material and a large area analysis, then you might expect the type of signal to noise that we see here. However, when we limit the availability of signal, then what we have to do is find ways to improve the sensitivity of an instrument or extend the acquisition time so that we can then try to obtain the type of signal that we can genuinely interpret using techniques such as peak fitting or curve fitting, as we're going to describe in this video. This is an example of a sample for which changes do occur to spectra, but the measurement that you see here is actually measured over a period of time that provides the type of signal to noise that we would like to see for this type of data. Now, the problem that we'll encounter when trying to analyze these data is that we have different oxidation states for copper that are within this average spectrum, averaged over time, averaged over analysis area and depth. This represents an average spectrum that we will have to peak fit and try and understand the different shapes that are within these data that we'll then interpret as chemical state. Now, if we measure the same sample under the same conditions, but this time, rather than aiming for good signal to noise in a spectrum, we take the same acquisition time and we aim for the same signal to noise over the entire experiment, even though we save the spectra as individual data that can be analysed en masse. And when we do this, instead of having a single spectrum for the copper 2p, we have many spectra, and when we look at these spectra, we can see that an evolution is occurring throughout the time period that corresponded to that previous measurement where we had a single average spectrum. Now, given data in this form, we have a clue as to what might be happening for this copper, and this clue may give us insight into how to fit these data and understand what's there. The fact that these data were evolving over the time period of the measurement when we had an average spectrum is not a bad thing. It's a bad thing if we don't know it's happening, but if we do know it's happening, it gives us the opportunity to try and work out from a data set the shapes that are within the data set that can be used to fit the data and can be interpreted as different chemical state. Here we have an example where the data set that we've just looked at has been analysed and two spectral shapes have been determined from those data, which are then fitted to each and every spectrum within the data set and produce results that you see here. Now, the method that is used to calculate these will be explained in what follows, but in this context we can see that shapes are available within the data set, and not only do they fit the data at the first scan, it also fits the last scan, and even a scan where there was some dropout in the signal, that by analysing the data set as a whole, even the anomaly that occurred in the data that we would prefer not to have happened can be compensated for, in the sense that we can fit this shape here. Even though poor signal to noise, we have shapes that are sympathetic to these data, and therefore they fit the data rather well. And when we go to the total spectrum, this is the spectrum that is the sum of all of the copper 2p spectra, the same two shapes fit this again. So we're able now to look at these shapes and try and interpret what we see. And certainly in this case you can see there's a satellite structure in one of these, so we might think that we've got some 2+, and there looks like there's not much of a satellite structure, so it's possible it's a reduced state rather than a 2+ state. Maybe it's a 1+, maybe it's there is some other explanation, but nevertheless we can see within the data there are two shapes. I keep referring to fitting these data, and what do I actually mean by fitting these data by these two curves? Well, in terms of linear least squares, what I'm doing is using a function that I calculate by defining an expression in terms of a set of curves. So in this example here I've got m curves, each one is represented by a function, and they're all scaled by these coefficients that are determined in a linear least squares sense. Now this is significant because the data has been used to calculate these shapes, so if done correctly then a linear least squares fit works very well. If we don't have shapes that we know are precisely from the data, then we tend to go to a non-linear least squares approach, and the non-linear least squares approach involves making use of functional forms, usually bell-shaped curves, Gaussian Lorentzians, Voigt functions, and these functional forms are then adjusted in terms of relative intensity using these A parameters, offset in terms of the B parameters, and the widths can adjust in terms of these C parameters, and these are adjusted to give us a function just as the linear least squares function provides a solution. Both model the data and allow us to reproduce the data by a synthetic envelope, and we test for the goodness of fit using an expression such as this. This is a combination of both a chi-square and a sum of squares depending on how you interpret the sigma I. If the sigma I are the square root of the data channels, then we're compensating for the pulse counting variation in each data channel, and we calculate a chi-square. If we set sigma equal to 1, then it's a sum of squares, and both of these may be used to assess whether or not we have a good fit to the data. Before looking at how we create curves from data, it's worth considering how we go about fitting a peak model to data. The same logic that is used to fit a peak model is inside the logic that we will use to create the line shapes. The difference is that we make assumptions in the case of a nonlinear optimization using bell-shaped curves. We assume that we have some understanding of what line shapes ought to look like. So in this case we've got a sample that is sucrose, and I'm going to use this to illustrate how to calculate curves from sucrose data. But if we look at this particular spectrum, this is the first spectrum in a sequence of spectra that were measured that I will subsequently use. But understanding how a peak model applies to this data might give some insight into what I'm actually going to do when I calculate these line shapes. So I'm starting off with an assumption that I have photoemission from carbon, and I can identify different chemical states within this molecule and introduce peaks that represent the signal from these different chemical states, and then I arrange them in a way that I think is appropriate for the spectrum given the context of the material I think I've got. So for example here, sucrose, if this is an accurate model of what sucrose ought to look like, and it is representative of the sample that I'm measuring, then I ought to see a set of carbon all with one chemical state, and they're all coloured blue here. They've all got the COH type structure, and so there should be a peak that represents the number of carbon atoms at a binding energy that represents the chemical state for carbon bonded to OH. And similarly we've got 2 that correspond to a CO type bond, and we've got two carbon atoms that correspond to an OCO type bond. So we arrange these components in this particular way and then we fit it to the data. But it's not as simple as that. We have to add additional components. For example, this green one, this would be usually explained as some contamination of the surface, and so this is a CCCH type peak. We have other structures here. Perhaps we could explain these in terms of the ring structures that we see in this representation of sucrose. So perhaps these are lost structures that we would anticipate being part of the spectrum, and so we justify a set of peaks and we introduce them and fit these peaks to the data using the chi-square or the RMS, using nonlinear least squares. So we adjust position, full width half maximum and area. But then if we do this without any constraints, we may get answers that don't make sense in the context of this material. So we introduce some constraints. We may introduce constraints that say there must be, in this case, there are 8 blue carbon atoms in a given chemical state compared to 2 and 2 in different chemical states. So we may look for a 4:1 ratio in these peaks. And that's what we have to do to get a meaningful fit that is purely guided by the figure of merit. The alternative to using a peak model and making assumptions about bell-shaped curves and how they relate to one another is to try and calculate shapes that derive directly from data. And this is an example of a data set that has been measured from sucrose. The data are sequentially measured so that we have 101 measurement cycles of carbon and oxygen. And within these measurement cycles, at least within the carbon 1s, you can see that an evolution in the data has occurred. So this gives an opportunity where a pair of spectra can be calculated from these evolving spectra. And the key point about these calculated spectra are that one looks very similar to what you would expect for sucrose in its raw state, and the other one, less obvious what it is, but nevertheless it has a classical shape that can be fitted with bell-shaped curves. So these both need explanation. One is easy to explain, the other is less easy to explain, but they both together are capable of reproducing what you see here in the carbon 1s spectra over the entire set of 101 measurements. And the way that these are calculated is based on looking at shapes that derive from 2 spectral forms. So I choose 2 spectra from the set of spectra, and I create a set of different spectra, and from these different spectra I identify shapes such as these. So the first question is, how do I know there are only 2 spectral shapes that can be used to reproduce all of the spectra in this experiment? The answer to how do I know there are only 2 significant shapes in this sucrose dataset lies in the mathematics of principal component analysis. And while principal component analysis, or PCA, makes use of linear algebra and the solution of eigenvalue problems, it can be explained in terms of a scatter plot and what we see in a scatter plot that is formed from spectra. In 2 dimensions, when we have 2 spectra, we can plot a set of points that are determined based on pairing up the data bins from these spectra and using the intensities as the coordinates along these coordinate axes plotted in 2 dimensions. And if we have 2 spectra that are identical, then background points from these 2 spectra will all cluster over here, and from the peak maximum we end up with a point up here. And so taking over the 2 spectra, the distribution of points extends along a line, and it extends along this line because these 2 spectra are identical. In other words, given 2 spectra, if they are identical, then rather than appearing as a 2-dimensional scatter plot, they appear as a 1-dimensional scatter plot, albeit at an angle. Now, if instead of having 2 identical spectra, we have two spectra that are slightly different, then there is a general direction or a line along which the scatter points appear to lie. So again, we have background points down here, peak maximum up here, and the general trend is along some particular line. And the fact that they are different means that instead of just being in one dimension, that the distribution then extends in two dimensions. So we can see that we have a two-dimensional scatter plot. Now what principal component analysis does for us is it calculates this line in the two-dimensional space that is defined by this scatter plot. This line represents the direction with the greatest variance of these data points, as we see plotted here. And the calculation that is performed by PCA gives us a mathematical tool for changing the spectra into new shapes, that if we do the same operation of plotting the distribution of these two shapes, we get the same shape as the original plot from these two spectra, but the plot now aligns with one of the axes. So this is the first abstract factor which has greatest variance and represents the direction of greatest significance within the scatter diagram, but is now aligned with the axes, and the perpendicular direction now gives you the variation that is the deviation from this direction of maximum variance. So the PCA is giving us a systematic way of understanding two spectra that we can see are close, but this shows us how close. One is very significant direction and the other is slightly different, and the big difference between these two spectral shapes that we see, the original spectra versus the PCA abstract factors, is that the abstract factors are orthogonal, which is why we see the alignment of the scatter plot as shown here. Now what does this mean if we have more spectra? So in the case of 3 spectra, this is a mathematical description of what we mean by a scatter plot, is that we have 3 sets of data, these D1, D2 and D3 vectors, and each one has a set of coordinates x1, x2, etc., y1, y2, etc. And these represent 3 spectra which, if we take the data bins and we create points using each one of these xi, yi, zi, we end up with a point in space that is part of an overall scatter diagram, that we can then determine a direction that is the maximum variance of the distribution. And we do this by calculating a sum of squares. This is the sigma I equals 1 to n pi, represents the perpendicular distance between a point in the scatter diagram and the line representing the greatest variance in the data. Now if we were to calculate the optimum values for this line, which are described by these parameters alpha, beta and gamma, and these are the direction cosines of a line in a 3-dimensional space, we can then calculate the optimum values for these alpha, beta and gamma based on minimizing this sum of squares. Now we have to do this by introducing a Lagrange multiplier to ensure that we maintain that we have a unit vector for this direction L, and then we need to optimize. In other words, we apply calculus, and calculus gives us a minimum or a maximum if we differentiate the function with respect to alpha, beta and gamma, and then set the derivatives, the partial derivatives, to 0. These would be the conditions for a minimum or a maximum. So if we differentiate and set to 0 the derivatives, then we end up with a system of equations which, if we apply basic algebra, we end up with a matrix description of the optimization problem of finding this line here, which is a covariance matrix in an eigenvalue problem, where the solution of the eigenvectors gives us the direction of the line that gives us the greatest variance in the data. So this is a nice example of how calculus translates into linear algebra. The mathematical logic that was applied in 3 dimensions equally applies in n dimensions. So if we have 101 spectra, then we can do the same calculation that involves a covariance matrix, and we can calculate abstract factors that give us a measure of the significance of different shapes to the overall 101 spectra. And this is a calculation of the first 6 abstract factors from the sucrose data, and we can see that we have one that is very significant. We also have one that is similar to the case where we had 2 spectra, and then we have another one. There are some shapes within these data, but what we can see is the order of magnitude of these different variances is changing from 10 to the 5, 10 to the 4, 10 to the 3, then it's 10 to the 3, 10 to the 3. So we have 2 that are significantly different from all the rest, and when we look at these look like noise associated with data. The shape you see here is typical of Poisson type noise, where the noise increases around a peak maximum. So it would be quite easy to identify these 3 as being noise. This one, not quite so easy, but it's of a similar order of magnitude to these. So on this basis, if we use 2 abstract factors, the ones that are orders of magnitude different from the ones we associate with noise, then we can calculate an approximation to the spectra that is based on these 2 curves here. And we'll perform the same linearly squares type calculation, form an approximation to the original data, and when we do this and apply that approximation to a set of spectra, these are just a subsample of the entire data set, we can see that the difference between the approximation and the raw data is reflected in the residual standard deviation and the residual itself. And you can see that in each one of these cases, starting from 0, 20, 40, up to 100, all these approximations are within the bounds that we would expect for pulse counted data. And we would probably accept residuals of this nature for nonlinear least squares fits to these data. So this shows that 2 abstract factors are giving a fairly good approximation to the data. So PCA is telling us we're working in a 2-dimensional space. Even though we have 101 spectra, they all lie in a plane. So we can choose 2 spectra out of the set of spectra, and they ought to fit the data set. But more importantly, we can choose 2 spectra from which we can calculate new shapes. And the new shapes are calculated using the concept of different spectra, which are formed by taking different proportions of these 2 spectra, subtracting one from the other, and observing the shapes that result. As part of this calculation, we will see spectra that look very much like sucrose, and that's an easy solution for us because we have an idea of what sucrose ought to look like. However, for the degraded form of sucrose, it's not clear what that spectrum ought to look like. So what I've done is I've plotted here a set of different spectra with different proportions of the 2 spectra we see here, and there's clearly a wide range of possible arrangements of photoemission peaks that could be associated with carbon chemistry that could contribute to what we see in degraded sucrose. So we have to make a choice, and we have to appreciate that that choice is not a unique choice, and we have to find some way of justifying it. So to justify a given choice, what we need to do is fit the 2 selected different spectra back to our original data and observe how well the data are reproduced by these 2 shapes, and whether these fits to data can be used to construct peak models that help us to understand the chemistry of sucrose when degraded by the analysis process. The example of sucrose was chosen because it was very simple. It was a 2-dimensional problem, and it illustrated the steps taken to calculate spectral shapes using different spectra. But the same technique, with a little bit more work, can be used to analyze data sets that contain 3 and 4 dimensions or more. It takes considerably more work the higher the dimensions we have, but nevertheless it is possible. And here's an example of a data set that is analysed by the same techniques applied to spectra that were measured from molybdenum trioxide when heated. And the idea is that we started with molybdenum trioxide and we think we're heading towards molybdenum dioxide, but how these two phases move between one and the other as a consequence of different temperatures, this has been highlighted by using this technique of calculating different spectra and then fitting your different spectra back to the original data. So in this case we actually have 4 different shapes. 2 of them derived from the initial modifications at a given temperature. So at 400 degrees Celsius the changes were characterized by these 2 shapes. When we go above to 450 degrees Celsius, then we end up with the original 2 shapes plus additional shapes that are required to fit these data. So this gives us some kind of handle by which we can understand how molybdenum trioxide in a thin film may transform into what we believe to be molybdenum dioxide.